From Milwaukee, Wisconsin, it's Wisconservation, a podcast that shines light on Wisconsin's conservation issues, research, and practices through thought-provoking conversations. I'm Brad Steckart, and today I'm here with Todd Levine, who's a lecturer from Carroll University. Today, Todd will share stories from his ongoing scientific pursuit, which took a much different path than he had imagined in his early 20s. Todd will explain how a simple lecture opened his eyes to an aquatic species that shows fascinating behaviors and is much more common than the large mammal species like polar bears and grizzly bears that he originally set out to study. Hey Todd, so what are we looking at here?
I've got a super nice picture that I've been using as my quintessential picture from 2015. I had a pair of students who did some great work in the Mugwanago.
Okay.
And I would love to get that picture slapped on it somewhere.
That would be sweet.
Yeah.
Okay.
Yeah, and it's just,
so
it's quintessential, right?
Yeah.
Like, you can see the river behind them, and for whatever dumb reason, they decided to do their research on a table.
Okay.
So they brought a folding table to the river, and they're decked out in like official Carroll shirts. They're not our colors, so they must have been like for a program or something.
Okay.
They're bright red instead of the orange.
Okay.
And so they've brought this folding table to the river. They're in the middle of a mussel survey, and they've got mussels. They're one of them's processing mussels, and the other one's collecting them. And the sky is just this perfect blue, and it's mirrored in the river, and you can see like classic riparian vegetation on both sides.
That is sweet.
It's like this is the quintessential Todd research picture.
That is like the ideal picture. It's like
Right.
They have the mussels there. They're
Right.
looking at the mussels on. They brought a table. That's actually for a student. Like, who thinks of that? Who thinks ahead that much?
I've
My
got bright
interns.
students.
Yeah. You do have bright students. I've met them.
Yeah, you've met a few, haven't you?
Yeah.
Yeah, so they would actually carry this thing down the river and occasionally would float it, but apparently the plastic wasn't totally sealed, so they got water in it. And then floating it was not an option.
Then they were sinking it.
Right.
That's awesome. I do want to know more about your mussel research, though. So, kind of just randomly finding a question on here.
Sure.
You research unionid mussels.
Right.
And am I saying that right? Unionid?
Yeah, unionids, Yep.
Unionids. So, what is a unionid mussel?
So they're commonly referred to as pearly mussels.
Okay.
There are a couple of families, but in North America, we've basically got two.
Okay.
So we've got the Unionidae and the Margatiferidae.
Okay.
And we don't have many margatiferid mussels at all. So in general, we're not just talking about the superfamily Unioidea. We're talking about the family Unionidae.
Okay.
I know that's a little taxonomic.
Just because you can pretty much generalize.
Well, it's a little bit easier to generalize within those families.
Yeah.
Like, they do some things that are really similar within the family.
Okay.
If that makes sense. So they're pearly. They've got these relatively heavy shells. They've got the inside called the nacre has kind of that.
The nacre.
That, like, right. No worries.
I don't know muscle morphology. So.
But it's kind of the classic mother of pearl.
Oh yeah, like
that kind of color and the opalescent.
So this is the kind of mussel that when you're boating around in Wisconsin, like, and I'm totally generalizing.
No,
here,
go for it.
But you're boating around in a Wisconsin lake and you look down and you see that big, kind of robust looking pearly kind of mussel. That's a unionid mussel.
Almost certainly.
Almost always. Okay.
Yeah.
Cool.
Yeah, so these are distinct from like the invasive species.
Okay.
Like the dracenids.
Right.
Like, really, really different. And
that's like a zebra mussel and quagga mussel.
Yep.
Okay.
Yep. Those are the dracenids.
Okay.
And they're in a separate family. It's actually more taxonomically different, but we probably don't need to get that nerdy.
Okay. We don't need to separate like what.
Right.
Yeah. Okay.
But some of the interesting things, like things that I'm fascinated by, are zebra mussels swim out. Their larvae like swim and drift.
Okay.
They're pretty weak swimmers, so they do a lot of drifting.
They kind of float, right?
Yeah.
Like a leaf.
Yeah, and the unionid mussels, and maybe I'm forcing the conversation to the cool part.
No, please do. Please force it straight to the cool part. I want to shoot
Cool.
the coolest stuff.
Good. So unionid mussels have this fascinating life history trait where they have larvae that attach to fish.
Okay.
And those larvae have to attach to fish for the most part. There have been some that people think, well, maybe this one doesn't have to attach to a fish. But when you look at the unionids, it's pretty clear that they're obligate parasites of fish.
But mussels don't swim, right?
Right.
I mean, that
Right.
much
Right.
relatively to a fish.
Yeah.
So these things that kind of sit still in the bottom of a lake bed or a river bed have to somehow find a way to get to fish.
Yeah, and I think maybe we can back up a little bit even. Like, so the zebra mussels, they've got some babies, they spew them out, they go do their thing, they drift on the currents and whatever.
Shoot them out.
Right? So
they're like your our selected species, right? Where they're just like tons of babies.
They've got lots of babies, and they're not, they don't have a bunch of ways to get upstream. They don't have a bunch of ways to get to new places.
Okay.
And that's where the unionids do something really cool.
What do they do?
So the unionids have to attach to a fish. And this means that, they can move from place to place, even upstream. And that's one of the reasons why that might be such a valuable thing for the unionids, is that a lot of the species are river species, right? So we have some in lakes. Mm-hmm. They're clearly in lakes, and you can go to lots of Wisconsin lakes and find unionids.
Totally.
But we've got lots of species in rivers, and that's a great way for them to get upstream. You attach to a fish.
That's amazing.
Their larvae can't really swim, but the fish can.
The fish can, yeah.
Yeah.
And a lot of fish go upstream to spawn a lot of the time,
right?
Right? Yeah.
Right. And I guess maybe at this point it would be good for me to say something about the number of species that we have in North America, because I'm going to get really self-conscious about being correct when I talk about these things.
Don't worry about it.
But we have 300 species in North America.
Of unionid
mussels.
Okay.
Right? So 300 species on the continent.
Wow.
Around 800, 900 globally. And then in Wisconsin, we have 51. I've seen recent estimates sitting at 52. But, just to kind of give you an idea of how much variety there is.
Yeah, that's kind of a lot.
And you were asking a minute ago about, okay, we've got these mussels that don't swim. They've got one muscular foot that, drags them
Yeah, they're
along.
not going fast.
They're not going
by any
fast.
means. Yeah.
No, they're going the length of one of our bodies in a week, 2 weeks,
yeah, not
right?
very fast.
Yeah, so they're not quick. So how are they getting to the fish? And that's what drew me in. I grew up on rivers. I grew up in just north of the Ozarks, and right where the Missouri, the Mississippi, and the Merrimack rivers meet near
Really?
St. Louis, Missouri. And
Wow.
and I loved rivers.
Yeah.
That was my lifeblood for a long time, for my whole life. Sometimes I can look at a map and tell where I am better by the rivers than by the highways.
Really?
There are some places.
That's pretty cool.
Yeah. And I thought that I was going to work with large mammals. I'd hooked up with a retired bear researcher.
Really?
And had actually done some work at his not-for-profit. Had tagged along on a trip to Churchill, Manitoba to watch the polar bear migration out onto the ice.
No way.
Yeah.
And you went and saw that?
Yeah. It was awesome.
So wait, this is early in your career, or this is
when I was an
career?
undergraduate.
Okay.
Yeah, before I decided to work on mussels.
Wow, that's amazing. So you were like, oh, okay, obviously mammals are so cool.
They are.
You want they're big and robust.
And they're like us.
And yeah,
and
exactly.
you can see them.
People relate to them. They're keystone species because people all want to it's easier to want to save something that's more like you.
Right. So they make these great conservation ambassadors.
Totally.
Some of them control ecosystem processes in the keystone species sense. Some of them are umbrella species because if you protect that one species, you can protect all these others.
Sure.
And yeah, I mean,
I didn't know
they're things that everybody knows.
Yeah.
You know?
Yeah.
So I've got this history with polar bears.
Interesting.
And we can talk about that later if you want.
Yeah, let's get back to that. I want to get back to that later because that's cool.
But so I'm working with Chuck Jonkel, one of the bear biologists from University of Montana, and volunteering at his not-for-profit. And I go with him on this epic trip to watch the polar bear migration out onto the ice. I was like, this is awesome.
Yeah.
This is great. And eventually I go to my very first professional conference, which is with wolves.
Okay.
Up in Duluth. And
that's awesome.
And that was cool,
Wolves are, yeah.
And I love wolves, I love bears, clearly large mammals. Yeah. And it's like all those things are awesome. And at some point I realized, everybody loves those critters. Everybody wants to know about those critters.
Yep. And
we can't ask basic questions. And so I was kind of thinking, like, I want to help them, but I don't know if there's, like, real... I don't know how much basic science. A lot of it's moving towards applied stuff. Yeah. And there's basic science to be done, right?
Yeah.
And people are doing that, but it's super competitive.
And what do you mean by basic versus applied?
Well, I'm thinking about things that help us to understand the fundamentals of how life works,
okay,
how populations work.
Okay.
Whereas, a lot of the work that I saw done or that I had access to was more about how many individuals do we have here? Or do we have a reproducing population in this?
So like applied to
range?
A certain area.
Yeah.
As opposed to like basic questions that you could ask like the world.
Yeah. Okay. And so I wanted to understand more about how life works.
Yeah.
And I wanted to get to that stuff, which I was really excited about.
Yeah, that's really cool.
And I loved the large mammals, and I wanted to do the work that was going to affect their conservation.
Yeah.
But at some point there was a tension sort of growing between I can't get to that basic science, but I can help with conservation, and that's cool. And eventually with Chuck's not-for-profit, the Great Bear Foundation, I went out and I spent a summer collecting samples of hair.
Okay.
From grizzly bears.
Which is common for mammal research, right? Like you said, hair traps.
Yeah, and not to date myself, but we didn't have a lot of genetic data at that point.
You can't be dating yourself that bad. You're not.
True. No, I don't have that many years on you yet.
I didn't think you did. Yeah, we're pretty close.
But we didn't have a lot of genetic data at that point, and it was really cool to go out and do that. And then I started asking questions about the bear hair stuff, and I got enough response that I knew what they were doing with it.
Okay. And what were they doing with it? Just seeing like
so they were
how many
looking,
individuals were in...
They were asking one of those quintessential management questions. And that was basically, do we have a reproducing population outside of a particular management area?
Okay.
Which has all sorts of legal ramifications too.
Yeah. I mean that's almost like it's almost an economy question.
Yeah.
And like almost a hunting question and all this other kind of like management.
And at that time it had a lot to do with which regulations applied to which part of the population.
Mm-hmm. Yeah.
And so that was the not-for-profit's interest in that answer.
Okay.
And I think that it makes sense, and I'm glad they're doing that.
It totally does, yeah.
And I'm glad I went and hiked around Montana to collect bear hair.
That's got to be like one of the greatest experiences as like
awesome.
An undergraduate. Just like, I'm here to collect hair. I'm like checking these traps.
Yep.
And it
was fantastic.
Yeah, it's amazing. It's very cool.
But then, I started asking questions about like, where's this hair going? What's happening with it?
Sure.
How do we know that they're all related? Or how do we know how they're related?
Or how are we testing if they're even related or not, right? And
I got some answers.
Okay.
But it made me really thirsty for more, and it made me think about how I could contribute to basic science. Because here's this whole burgeoning field of genetics, right? And yeah, we've had genetics for decades.
Right.
But at this point, we're starting to apply a lot of the stuff that we've learned about genetics to conservation.
Right. I mean, when genetics started, it was focused just on humans. I mean, because we wanted to know about us and we wanted to know about like our lineages, and I mean it was like plants and stuff like that, but people wanted to answer human questions.
We certainly did, and there's been the focus on things like the Human Genome Project.
Right.
It's really important. It's important to health. It's important to a lot of basic and applied questions there too, right?
But at this point, I think people were starting to go towards being able to use this for conservation.
Yeah, and we were starting. Journals like Molecular Ecology were relatively new.
Yeah.
And we were starting to see all the different ways that we can bring this into the parts of biology that I wanted to engage in. But I wasn't getting real far with the questions.
Right.
So I was also a member of the Tri Beta National Biological Honor Society.
Okay.
Which, is neither here nor there, but they had this really cool program where they did a retreat every year. And I was like, oh, I get to get out of town?
Sounds awesome.
I get to leave it all behind.
Sure,
and we go up to a field station, and it would be fun, and
Sweet. Learn some stuff.
Have an awesome weekend.
Yeah.
And there was a keynote speaker each year.
Okay.
And the keynote speaker that one pivotal year.
The pivotal year for
for
Todd.
me.
Yeah.
Was Chris Barnhart from Missouri State University. And Chris talked about these mussels that were hanging out, and by the way, they're right here in the Ozarks. So I've been thinking about like how I needed to go to Churchill to look at polar bears, or maybe I'd go to Central America and work on some of the big cat species, or whatever, something like that. And I was thinking about how I wanted to work with these big mammals, and then Chris comes in and talks about these mussels who are catching fish. And you're like, what? Exactly.
Mussels that are catching fish?
Yeah.
What?
and I've now spent, 20 formative years canoeing. I guess I was just canoeing at that time. But, floating over these things, and I had no idea this was happening.
Yeah.
And then he starts throwing out numbers like how many species are there? Like, I thought there were 2.
How would you even know, right? If you're not looking at it.
Right. I mean, it was really, it was super eye-opening. So at that point, I was starting to look for grad schools and eventually found an opportunity to work both with genetics and with mussels.
Okay.
And so that's also one of my first professional connections to Wisconsin.
Really?
Is I'd wanted to go to school here, and I'd looked at it, and for logistical reasons it didn't work, and both
pretty far from your home,
and
right?
Grad.
Yeah.
I mean, it's not so bad. It's upriver.
A lot of things are upriver.
Good point. And Chicago's upriver now.
Yeah, right.
Used to be in another watershed, but
what?
The connection, the canals.
Oh, just really?
Yeah. So
just that recently?
I mean, we built a canal between.
I mean, I just
the Great Lakes
know.
and the Upper Mississippi River.
Yeah, I guess the Chicago River, right?
I think is the I&M the first one.
I don't know.
Anyway,
I should know this.
There's a series of canals. Yeah, you should. And so should I.
That's okay.
And a fantastic reading recommendation is Life and Death of the Great Lakes.
Yes, I just bought that book. Yesterday.
And they just had the author in Madison, I think.
Dan Egan.
Yeah.
He's a I've YouTubed him.
Yeah.
But yeah.
So my aquatic ecology class this year just went and checked out some of the canals and did a watershed field trip.
Really?
Like an overnight field trip where we drove down the Fox to the Illinois, checked out the canals, and then went down to Starved Rock and looked at like glacial and geological history.
Dang, that's sweet.
And like wrapped it in.
Really?
You should come with us if we do it again.
I will, yeah.
Cool.
Just let me know next time. I'm in. That sounds really fun.
It was awesome. But anyway, so yeah, we built canals and now Chicago's upriver of St. Louis too.
Okay, I got you.
Anyway,
I follow.
Yeah, so Chris Barnhart did this really cool talk on these random these amazing things that mussels were doing to live out their lives, and I got an opportunity to study the genetics of mussels, and I looked at post-glacial genetics of freshwater mussels in North America. And for a whole variety of reasons, I kind of moved away from that during the project, and started doing a little bit more with demography and population estimation and things like that. Kind of back to some of those applied questions.
So is that because, and I feel like this happens a lot of the time with people who are doing research, and it's an awesome thing that happens, but you start wanting to answer those basic questions, and you find that you answer them. Like you find that you're like, wow, like I did my research and I'm answering these questions. And then all of a sudden you're like, but what about a population? Because this population's unique because of this, and then this population is unique. Right. So you
although I have to admit that some of it was logistical too.
Okay.
Like we had funding to do population sizes.
Okay.
We didn't have funding to do population genetics.
Okay. Well, there you go. That's
and population sizes cost a lot less.
Really?
Actually.
Yeah. Okay. Well, genetics are expensive, right?
Yeah. I mean, so I had field time for both.
Yeah.
But then, one, I sat at a computer and work. The other one, I'd now have to bring back to the lab and go through all those steps.
Mm-hmm.
So it's logistical stuff, but it was also interest. I ended up completing both projects, but I've got stuff published from the field work and the population work, and I never. I'm one of those bad PhD students who never published a chapter.
That's not being a bad PhD student. That's just not.
It depends on who you ask.
Yeah, well, that depends on it depends on your advisor.
But we're going to come back to that in a little bit.
I want to ask though, because you explained that you heard this really kind of eye-opening keynote speaker, right?
Yeah.
What was his name?
Chris Barnhart.
And you said he was these mussels, he said, were attracting fish.
Yeah.
But we haven't talked about how that happens.
Oh, that's a really good point. Yeah,
so I want to know like how do these mussels, because so there's a couple things going on in our conversation. One is zebra mussels and quagga mussels, who they just spurt out these floating veligers or babies that just kind of float around, they latch onto something and they're fine. They're going to do okay.
And when you say latch onto, I mean we're talking about they're eventually changing and getting that adult form and attaching with byssal threads like they're stuck.
Right. So they're floating around. So this is the invasive zebra mussels. They're floating around, kind of wafting, and as they're doing that, they're collecting calcium and starting to build their shell, I guess. And then as they do that, they kind of get heavy enough, right, where they kind of fall onto a hard surface.
Yeah.
Is that pretty much what happens, more or less?
Gotta be honest, I don't remember a lot about the sort of metamorphosis there.
I think that's kind of what happens.
That
they land on a plant
reasonable.
stem or a boat or a pier, and then they start to kind of grow, and then they grow the rest of their shell and start to eat, and then that's kind of the story of the zebra mussel. It kind of sits there for a while.
Yeah, and they're they're doing filter feeding just like our native mussels.
Right.
But there's some differences there too.
But the other thing that happens is our native mussels, which are totally different. I mean, they've evolved in a totally different way. And I guess I just don't really know what it is. Like I know a little bit, but if you could explain it, that would be awesome.
Yeah, I'd love to. That's literally what Chris Barnhart told me and told everybody who was in that audience. That changed the course of my career, right? And basically what mussels do is they have this whole suite of adaptations. All of them are aimed at getting fish to come into contact with their babies. Okay. With their glochidial larvae. Okay. And we should get that out of the way because I'm going to say glochidia.
Glochidia?
Glochidia.
Okay. Yep. So is that different? Okay.
So that's the larvae. That's the larvae of the unionid mussels. Okay.
Glochidia.
Yep. And these guys, they've kind of got this bivalve shape, but it's very basic. Okay. They're sort of a very simple version, and we could talk about more distinct differences, but I think that gives you an idea, right?
So they're kind of like the cartoon clam, where they have like
exactly
the one shell underneath
I like the way you
and
said
the one
that.
shell over, and they have a hinge, right?
Yeah, so they've got two shells.
Yeah.
And they sort of clamp.
Yeah.
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 And if you look on the internet, you can see like wonderful videos of when you add salt to a petri dish of larvae, and you can see them clap.
Really?
Uh-huh.
Because they get...
so the idea is that that's probably a chemosensory thing where they're like, oh, look, it's salt. Maybe there's a host here. And if they get a hold of some tissue, they clamp on and here's their host.
Oh my God, that's so cool.
And there's probably more going on
because and the reason like fish or whatever has like salt in their blood or salt, like they produce saline. So that's like an indicator that's so cool. That's amazing. Yeah.
Okay. So these guys are just amazing. You know, one of the videos that's out there has, talks about how they got these elaborate displays that some of them do, and they don't have eyes.
What do you mean by displays?
So yeah, that's probably the quintessential like unionid thing to talk about.
Okay.
Is that there are lures that the moms grow out of their own bodies. Lures, yeah.
Like fishing lure.
Yes.
Okay.
Exactly like a fishing lure, their own flesh. Literally their own flesh. So the moms brood their glochidia, brood their larvae in their gills.
Okay. And
the gills kind of get big and distended and kind of squished out. So they're more vulnerable. They're so you can actually see them
like outside of their shell. So they're sticking out of their shell. So if something came by, I could bite it off.
Yeah, but they've also got this tissue on the inside of their shells called the mantle tissue.
Okay.
And the mantle tissue actually grows and expands beyond the shell margin.
Okay.
And that mantle tissue is what forms the lure.
Interesting.
And those lures can be super simple. So we have a genus of mussels that's common in Wisconsin, the quadrulas.
Okay.
And the taxonomy's changed a little bit, but this group uses things like catfish. And they live in really turbid waters in a lot of cases. They can be in some of the clearer waters, but these guys, these mussels use some really simple lures. And it's only been in the last few years that people, actually some folks with the Minnesota DNR and a couple of colleges in Minnesota recently described them. And these quadrulas use a pretty simple sort of squishy, fleshy lure.
Like a little worm?
No, it's just maybe like a little bit of another fish drifting downstream.
Okay,
like if you think about a little bit of fish flesh that's been bouncing along the bottom, looks a lot like that.
Okay.
And there's a hypothesis that maybe quadrulas use more of a chemical lure, like they smell like something.
So they actually...
We don't know that.
You don't know.
We don't know it. It's a hypothesis that needs to be tested.
Really? Oh,
yeah.
Wow,
I tried to test it and failed once.
We'll have to get back to that. Okay, hold on.
It's not that good a story, really.
Okay, wow.
But it was a good hypothesis that I failed to test.
It is a good hypothesis. That makes
Anyway, but it could be a cool story, and I hope that I or somebody else gets a chance to check it out soon.
Yeah.
But the visual lures, the mantle tissue part of those are really simple. On the other hand, some of the well-known lures, the iconic lures, I say iconic, but iconic to a small number of people. Right?
Well, I've seen the videos of these
right.
lures, and it's amazing because if you Google unionid mussel lures, you will see like these. It literally looks like a little fish,
right.
Like a little minnow, or like a little,
Yeah, so
it's amazing.
So some of them are pretty basic. And I'll try to get you some video later of some of the basic ones.
Yeah.
I did some behavioral work a few years ago in 2014 with one of my students from UW Rock County, now UW Whitewater at Rock County.
Okay.
In any case, she went on to UW Milwaukee, graduated, and now works for the DNR and the Coast Guard, which is really cool.
Really?
I'm really proud of her.
What's her name?
Erin Cox.
I know Erin. Yeah, I've met her. Of course you do. I've met her through. She worked for Heidi Bunk, who's the
you met her independently.
Southeastern Wisconsin lakes biologist.
Awesome.
Yeah.
I'm super proud of her.
She's a bodybuilder, right?
Fighter?
Fighter.
I think
yeah,
she's doing
fighter
MMA now.
Yeah, she's doing a bunch of like awesome stuff. Yeah.
Yeah, right before she went into the Coast Guard, she was starting to do a bunch of MMA stuff.
Yeah. She came out for a day and
She's one of my
for Starry
favorites.
Stonewort with me. Yeah, she was really cool.
And in fact, she went and presented the work that I'm talking about at a conference used to be called National Association NABT. North American Benthological Society.
Okay.
Sorry.
No worries.
Now Society for Freshwater Sciences.
Oh, okay.
One of the first meetings where it was actually called that. In any case, she worked in the Mukwonago River with me, and we've got some videos that we can share later that have what we referred to in a sort of colloquial sense as blobby lures.
Blobby.
Yeah, these things are just like these gray masses that the moms were producing.
Really? Just
Yeah.
like a blob of gray, just flesh.
Yeah, well, it's gray flesh that she's grown out of her mantle tissue, and it's kind of hanging around,
swaying in the breeze. swaying
Yeah,
in the
and so
stream,
there's
yeah.
not a lot of pigmentation,
Okay.
right? And the pigments are where I'm going here.
Okay.
So it's sort of this, this gray mass with a couple of blobs of white and black on it.
Okay. Well, so she's doing something.
Yeah, she's doing something, right? She made an effort.
She's trying.
Her poor babies may or may
Yeah,
not
yeah.
find a fish, but then we found these blobby lures next to some really intense and complex lures. So there are some that have these little spots of pigment that look like old-fashioned newsprint,
Okay.
like the tiny little dots.
Yeah.
That make up a pattern.
Yeah.
And so we found those, and I found those all throughout southeastern Wisconsin.
And right next, but these two species were right next to each other?
Well, these are the same species.
Oh.
So based on shell morphology and everything we know,
wow.
so we're going to get there in just a minute.
Wow. Okay.
But based on shell morphology, everything else we know about them, same species.
Okay.
And we found those next to each other in the Mukwonago. And then we found another lure type next to them in the Mukwonago. So this lure type has stripes.
Okay.
Like the pigments are super clear, like tiger stripes.
Okay.
And so all three of these lure types, same species of mussel, same river.
What?
So they're totally different. But the moms are all using them to imitate fish. So they've got a very distinct tail that actually separates from the rest of the mantle tissue and like twists up.
That's amazing.
Yeah. And so you've got this narrow fish body, and you've got this wider tail, and there's usually a separation between the rest of her mantle tissue and that little tail that's like free floating in the river.
So this is a species of mussel who literally don't have eyes,
Right.
has never seen a fish in its life,
Right.
and somehow is creating a little thing, like a little fake lure, that looks just like some sort of fish,
Right.
and has an actual like body section and tail section.
Yep.
How does that even happen? Like what?
Well, and some people might say at this point, this is where evolution's BS, right?
Sure, yeah.
And I get that.
Yeah,
I get that response.
That's like
right?
that's my first reaction.
This is
just-
amazing.
Yeah.
And it's hard to think about that in an evolutionary context. But I think that looking at those three types of lures, we've got these real general blobby ones, and then we've got those in another group, in another genus. And they're pretty simple and blobby too. Right?
Mm-hmm.
But then we've got these really complex ones.
These striped, like, cool tiger lures.
Yeah, and in other places, they actually have eye spots, like with white and then black.
So they actually have, like, determined where the eye is supposed to be on this.
Uh-huh.
Okay, and this is the same species?
Same genus at this point.
Or same genus. Okay.
So we're talking about Lampsillus for the one set.
Okay.
And then we're talking about another, the ones that always have the blobby lures, at least as far as we know, are Quadrulus. That's another genus.
Okay.
But these Lampsillus, like they've got some that are blobby. Mm-hmm. They've got some that are pretty complex with set pigmentation patterns. And then they've got these others that are super complex that have eye spots and have stripes like.
What's going on? Like these smart fucking.
Well, I think
right. How did they do that? How did they get the spot?
So the one way to think of it is, we've got this whole series of different mussels, and some of them live in turbid waters, right?
Okay.
Water that you couldn't see through.
Right. Oh, I see where you're going.
I love that dawning.
Oh, that's so cool. Okay, yeah.
But then
keep explaining.
We get some of these. So I was saying that I got my start in the Ozarks, and when you go out to the Mississippi River down there, it's muddy. It's chocolate chalk.
Yeah.
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 You go upstream, and it doesn't take very far upstream, and it's crystal clear. One of the things that may be going on here is that you've got a bunch of pigmentation patterns, and we got this blobby mussel, does okay where the water's chocolate sometimes.
Sure.
You've got this one that's really clear, but it doesn't look like a fish. Maybe that's a dead end. Maybe that one doesn't ever find its fish.
Yeah.
And these others that have these stripy lures, fish look at that. Ooh, that looks tasty.
Yeah.
Never had one of those before.
That's a morsel. Yeah.
Let's
right?
eat that.
Yeah. Let's try
right?
this out.
I mean, and so right. I mean, this is a hypothesis. It's a paradigm in some
So this
ways.
hasn't been proven yet?
I mean, we don't know whether that's the way that these lure types relate to one another or not.
Yeah, evolved, or how they,
Yeah. But, shameless plug.
Yeah, please, plug away.
I'm looking for funding opportunities. We'd like to look at these different lure types and ask whether or not there are unrelated genes that are the same or different.
Interesting.
So that's one of the things that I'd like to be able to give my students is to be able to take data on these genes and ask whether or not mussels in the same river have different genetic structures.
Wow.
Or different genes that are not related to the lure itself. Right? So we could do some interesting things asking whether or not these populations are actually separated within the same river.
That's very interesting.
I think it is.
Yeah, no.
I think a lot of people might not. But...
No, so you're asking, basically, are these mussels that are like the blobby? I'm going to use the blobby.
Yeah, go ahead.
Terminology. So these blobby
I've been told
lures,
it's not professional, but
that's okay. We're not.
I'm glad
we don't
you
need
like
to be
it.
professional here. So the blobby lures.
Yeah.
And the clear lures, let's say, I call them clear lures, may have like different similarities or differences in their genetics, which may determine that they're like. But are they determined right now as different species?
Nope. These are, when we look at the shell morphology, and really in mussels, for the most part, we're looking at shells.
Okay.
We're looking at shell.
Well, when you
shape.
okay, let's be honest. Okay. When you find a mussel shell, you're not finding the mussel itself, because usually when you find a mussel, like when I find a mussel in a lake, I never find a live mussel. I always find the mussels.
I'm sorry to hear that.
Well,
we need to get out of the field together.
Yeah, seriously. But I always find the ones that are opened up because they've died and they're all shiny and they're like pearly colored, and I find them and I think, oh, this is really pretty, and this is what people used to make buttons out of. Right, That's what I think of.
And you're right.
But I don't think of, wow, this must have had a really unique lure to attract other fish. Which, by the way, we should finish explaining. Because the lure is to attract a fish. Right. So what happens then? Because we got to finish the original story about how. So because we were talking about zebra mussels, they don't need a fish host. Right. But native mussels do need a fish host. Right. And how does that happen once they produce this lure? Right.
So the mussels attract a fish, and some of them have really complex behaviors associated with it. So the
mussels?
Yeah.
Okay.
Yeah, so the mom will flip that bit of mantle tissue in a way that looks like a fish swimming.
What?
Oh, yeah.
So not only the genetics, but the behavior that goes along with it.
So remind me, this you should write down.
Yeah.
We should come back to the rainbow mussel in a little bit.
Okay. All right.
But for right now, let's stick on this one. But yeah, so they actually have these behaviors where they take that mantle tissue and they're flipping it out so it looks like a fish swimming through the water.
Wow.
Yeah.
We'll have to add a video of that happening to this.
We got this.
Yeah.
We got this.
Yeah.
I spent a lot of time laying with my face down in the river and calling it a good day. That's why you gave me beer first, isn't it?
I love that. Yes, exactly. Cheers to that. So you're sitting in the river, laying face down, watching these mussels flap.
Yeah. I'm a really exciting person, right?
And man, it's more exciting than a lot of other jobs.
I've done most of those jobs.
So what does that do though for fish?
Yeah.
So they're looking like fish.
Right. So they're looking like
they're
fish,
baiting
and
fish
they've got predatory fish come in, take a bite of mom.
Okay.
And yeah,
so mom's asking for a bite.
Oh,
She's
yeah.
like, give me a bite of that northern.
Yeah.
I don't know why I turned into a southern accent.
Right. Your fish have an interesting accent.
I listened to some country music today. I don't know.
But yeah, they're asking for it, and the fish comes in, bites.
Yeah.
And they release their glochidia.
So as it bites, this
and
mussel senses that.
There's differences among species, and I want to be clear about that. And there may be some things that I'm not catching here too.
Okay.
So sometimes the fish is actually rupturing the gills. And that's releasing it.
So it rips a hole where the glochidia are kind of sitting, and then the glochidia as it's
sort of flush out.
Yeah, they just we're free.
In other cases, and I've got some other examples of ways that mussels catch fish in a few minutes, but they're amazing. Like they actually get a hold of the fish and pump their glochidia out.
Wait, the mussel grabs on?
Yeah.
Wow.
Yeah, and
so it's open, and the fish comes and bites it, and then it just...
Yeah, and we've known about the lures for about a century and a half, I think, or some of the earliest papers I've seen.
Really? Wow.
So when I'm looking back for historical references, I think the earliest ones are in the early 1900s.
Okay.
So we're talking button boom time.
Okay.
Right? North American button boom time.
Yeah.
But the head grabbing ones, we've known about for 12 years, 13 years.
What?
Yeah.
So 2006?
Yeah.
What?
Yeah.
Or 2000 somewhere.
Don't do the math real
early
careful,
2000s.
because I'm not sure
early 2000s,
which year it was.
But past 2000.
Yeah.
So it was the 21st century.
Yeah.
This is a 21st century discovery.
Correct. Yep.
That mussels actually grabbed fish.
The first time I ever heard
the mamas.
about it.
The mamas grabbed fish. Because we knew that the little ones bit with salt.
Right. The little ones grab fish, and the mamas just attracted them.
Yeah.
Right?
Yeah.
But then, and this was one of the people who was working with Chris Barnhart, and they got a video of this. And they're up on YouTube. You can find them.
Interesting.
But these are, these fish wind up going around, and they've got these the mussels have these bright white mantle tissues.
Yeah.
But they don't have lures the way that the ones I'm telling you about do.
Really?
They've just got a bright white sort of mantle tissue. And
do they
the
flap
fish
it?
kind of, not that I can tell.
Really?
Not from the videos I've seen. I've never seen this in person.
Okay.
I've only seen it in videos.
Okay.
I saw it at professional meetings, saw it on YouTube videos.
Todd's Saturday nights. Sorry. Whoa, did you see the glochidia get released on that one?
Right.
I can't believe that.
Right.
That's amazing, though.
But yeah, so these fish are pushing their way through rocks, looking for prey.
Oh, sure.
And they're sort of nosing. They have this nose pushing behavior.
Okay.
And they nose into these mussels, and she just snaps down.
Wow.
And then in the video, you can see the glochidia just pump past its face.
Like a little spurt.
Yeah.
Wow.
In just little.
Oh my.
And those are the unlucky glochidia, right? Those are the ones that are floating downstream to die.
Oh shit! Yeah.
Yeah. Miss the boat. Miss the fish.
Miss
whatever.
the fish on that one. That reminds me of I'm gonna go on a tiny tangent.
Yeah, it's okay.
But no,
because we haven't been on a tangent before.
No, exactly. It reminds me of, I remember in ecology class in college, Dr. Tracy Feldman, he took a tuning fork and he held it up to a tomato plant. I don't know if you've ever heard of this, but bees, they actually hum at a certain frequency. Really? So when they land on a flower, their inner muscles are still working at a certain frequency. So they make a sound, whether you can actually auditorily hear it or not. Okay. They're vibrating at a certain frequency. So if you hold a tuning fork up to a tomato flower, it'll actually like release.
Oh, that's
release
super
pollen.
cool.
Yeah.
Now I get where that's going.
It'll shoot out pollen. And I remember he had this tomato plant in class, and he's like, "All right, class, come on, gather around." And we all gathered around this table, and he had a tuning fork, and we were like, "What is this guy doing?" Right. But yeah, he hit it and held it up, and each flower, when he held it up to it, would just, like release all of its pollen at once.
Nature's cool.
Nature is awesome. So that's just what it reminded me of. I don't know if like anyone could ever relate those two things, but
you can.
Kind of sounds like that's what happens. Is they just kind of they get this sensory overload, and then they just like kind of.
Yeah, and in this species, I don't know if it's as similar, but you can take water that a fish has been swimming in, and you might be able to elicit a response in some mussels.
Really?
Right? So
without the fish being there.
Yeah, and so I guess you got to kind of throw smell in quotes.
Yeah.
But the mussels can kind of smell the fish.
They can detect.
Yeah.
They can't smell.
They have a chemosensory reaction.
Yeah, there's some receptor somewhere.
Yeah, and so I think that's another avenue that I think is ripe for behavioral ecology and freshwater mussels.
That's amazing.
Like, behavioral ecologists sort of bounce over these guys because, they're sort of living rocks. They filter stuff.
Right.
But my God, they've got stuff going on, And I think there's a lot of opportunities there, and I hope my students get a chance to look at some of those things. But going back to the lures, I mentioned earlier, and you're totally gonna have to splice this stuff together later.
No worries. We don't have to. I totally went down the wrong pipe.
I'm sorry to hear that. So it makes me feel a lot.
This should probably just be unedited. Just like.
Yeah, you think so?
Yeah.
Oh God. God help your viewers.
I'm sorry.
But I want to go back to these other, like you were impressed by the idea that there's an eye spot on some of these.
Totally.
Like the white and the black.
Yeah.
And some of them have lateral stripes, like one species of fish, and other of them have these vertical tiger stripes that, to my eye, don't look like a lot of fish that we have here.
But it's like a different pattern.
It's a very clear, distinct pattern, and it's set.
And it's distinct. Oh, in each one.
There are individual mussels every time we see them. They've got the tiger stripe lure out.
Really?
Yeah.
So it's not a unique by mussel thing. It's like one. Like a lot of mussels have similar patterns.
Yeah, there are a bunch of mussels that, and it's not a large proportion of the population. I think. If I'm thinking back right, it's like 5%.
Okay.
Or something. So, we surveyed a couple hundred mussels, and we've got 5, 10, 15, 20, right?
Right.
Something in
there.
But
of this.
But it's always this clear, these have stripes, these have newspaper pigment.
Interesting. Yeah.
Yeah.
Yeah, very interesting.
So that set of lures in the Lampsilus genus is cool. And it's really interesting. But then we've got these rainbow mussels.
Oh, yeah.
The Velosa iris. So those have conservation status in Wisconsin. When we talk about like globally or even nationally, they don't have the protections. But in Wisconsin, they've got conservation status.
Okay. Wait, how in Wisconsin, but not anywhere else?
At the state level.
Okay.
And let's...
So DNR, so the Wisconsin DNR, Department of Natural Resources, has determined these as an important species.
At a species that's in danger.
In danger. Interesting.
a species that's worth protecting.
And so
Right.
enough research has been done on rainbow mussels. Well, I guess with a name like rainbow mussels, you must have to explain you got to explain why they're called rainbow mussels.
They have these really beautiful green stripes on them.
Okay.
And I assume that has something to do with the name.
So that probably caught somebody's eye at some point who thought, this seems like something that I should keep track of.
Maybe. I mean, it's hard to know. It's hard to know, like where it got, where people got interested in it.
But like people aren't like, oh, tiger stripe mussels are, they're not.
Yeah,
like they're not
yeah.
like, well, in
No, I got you.
Yeah.
Let's see. I'm trying to find the status of these mussels that I'm talking about right now.
Of the rainbow mussels?
Yeah. Yep. Velosa iris. And the state rank looks like it's an S1, which I think is state endangered. So protected by state law.
Okay.
State status. It's state endangered.
So if I were to if I lived on a river, this is a random anecdote. So like if I were living on a river and my river was filling in and I was like, hey, I need to dredge my river because I want to take my boat out, and then they found rainbow mussels, they would be like, oh, hold up,
You might need to be.
you need to buy an
careful
extra
now.
permit
Yeah.
or like do something special for these mussels.
Right. So there's federal laws that protect.
If you want another beer, there's
Appreciated.
yeah.
I may wait a minute, but
Yeah, that's
but yeah, so there are federal laws, right? The Federal Endangered Species Act.
Yeah.
But then each state has different laws that protect different groups of organisms.
Sure.
Which
Rightly so, because so
Right.
many different states have different organisms.
Right, and I
You
guess
need to-
when we talk about the rainbow mussel, like this is a mussel that's doing okay in some parts of its range,
Okay.
but we're at the edge.
Oh, okay.
And so in some ways that puts us at the forefront of conservation.
Northern edge?
Like northwestern edge.
Okay. Oh,
so
wow.
they go further north to the east of us.
Okay.
And they go further west to the south of us. So we're sort of at the northwestern edge.
Interesting.
Of it.
Okay. Very interesting.
But that puts us at the forefront of conservation because there's sort of that means that their range, if something's going on, that means at the center of their range they might be doing fine.
Yeah.
But here, they're losing ground.
Right.
Right, and so the more that stuff happens, the more they're pushed back towards the center of their range.
Mm-hmm.
And that's something we need to be cognizant about.
Totally.
And that's, one of the things that I'm really proud about, the little bit of genetic data that I collected in grad school helped to inform some of the decisions that Canada made about their mussels. Basically, one of the things that they did is I'd worked with some Canadian researchers, and they sent us tissue samples. I sent them gene sequences back. And they said, look, we don't have a lot of genetic diversity. You've got really common mussels that we're looking at, but they don't have a lot of genetic diversity, so we should protect them at the edge of their range. Give them the best chance we can to survive. And in some ways, the rainbow mussel is like that, right? So it's native to the southeastern corner of Wisconsin. The southeastern corner of Wisconsin is at the northwestern edge of its range, which means that if we want to protect the rainbow in Indiana, if we want to protect the rainbow in Wisconsin, in Illinois, if we want to think about, the status in Michigan or Missouri, we want to think about what's happening here at the very edge of its range.
Definitely. Yeah.
And so we need to think about what's happening with these mussels right here, even though they're common elsewhere. We need to do something to make sure that they're not losing ground.
Well, it's almost like if something were to happen here and that was something that, affected their population, that's an implication that would ruin their population somewhere else.
Right.
So, yeah. So, I mean, that makes total sense. And it's like that happens so many places where, and that's one of those cases where like all of these species, they just don't they don't have like state or county or any kind of boundaries, right?
Right. So we put these geopolitical boundaries on top of them.
Right.
And
then we have different statuses for them in each geopolitical boundary.
And Illinois is different than Wisconsin, and Kentucky is different than Illinois.
Right.
But they're all part of its range.
Yeah.
And so we don't have this holistic approach to protecting species.
Right.
And the best we can do is something like International Union for the Conservation of Nature or Federal Endangered Species Listing. But we don't have some of these shared things that are independent of politics, right? This is a human boundary, not a natural one.
The politics are a human boundary.
Yeah.
Right.
Yeah.
Right. Well, and the state and whatever.
Right.
The political boundaries. Right.
Yep, exactly.
Yep.
So whether Illinois or Wisconsin is managing a rainbow mussel, it is completely human.
But
it has nothing to do with them.
But I feel like, so there are some boundaries, like the Fox River watershed.
Yeah.
Who goes from Wisconsin to Illinois. And I don't...
And that partnership, like the initiative to connect people in the Fox watershed is one of the most brilliant things that I've seen done.
Yeah.
Like, this is a group of people who are working together to try to manage this natural unit. And they said, it doesn't matter what geopolitical boundary we're in. It doesn't matter if we're Illinois. It doesn't matter if we're Waukesha County. It doesn't matter if we're Wisconsin. We're going to work together because we're dealing with this natural system that's part of all these.
Yeah. It does matter that Packer fans are better than Bears fans. That does matter. But that's the only part of it.
I mean,
really the only part of it.
If you feel that way.
I do. I strongly feel that way.
I'm not even surprised.
I hope you probably don't feel the same, do you? Oh no!
I didn't say that. I didn't say
that.
Let's not go there.
Yeah, let's not go there. You're not a Bears fan, though, right?
No.
Okay.
You're safe. We're safe.
All right. But no, it's true. I mean, they are like. There is no boundary that should.
Right. It's
really
the natural boundary
isolate.
That should decide
what's happening. Yeah, it's the natural boundary. And
yeah, we shouldn't let state boundaries. We shouldn't let city boundaries. We shouldn't let county boundaries determine how we deal with our natural resources.
Yeah.
The natural resources have their own natural boundaries, right? I mean, terrestrial ecosystems have continents.
Right.
Watersheds.
Well, and they have like latitudes and longitudes that we're like, all right, seasons, affect the land more towards this area. And
yeah. Yeah, and so we need to think about the natural boundaries rather than the ones that we've imposed on nature.
Yeah. Yeah.
But moving back to the rainbow mussel, so we've got this unique thing at the edge of its range. We're trying to protect it, and these guys have probably the most complicated lures of any of them that I can come up with off the top of my head.
Really?
They imitate crayfish.
What?
Yeah.
No way.
They have little appendages on the edge of that mantle tissue that grows out, and they look like crayfish legs.
What?
Yeah.
That's amazing.
It is.
So fish come up to them wanting to eat a crayfish.
Right.
And do they latch on?
They bite. No. These are the lures just like the fish lures that I was talking about earlier.
Okay.
Fish bites it, releases Glochidia.
Yep.
And fish swims away infected.
So by infected, I just want to make sure.
Sure.
What I know is happening. So infected means that the fish has in its gills the Glochidia latch on.
And I've heard that there's a better term. Some people argue that infested is a better term because these are parasites that are living on the outside of the organism, but then the organism's building up. They have a physiological response where they're building up tissue around the larvae.
The fish do.
Mm-hmm.
So they have like an autoimmune.
It's insistent.
Like if you were to get a cold and your white blood cells attack the bacteria in your body.
Yeah, so they're building up tissue around it, and so it's insisting.
So they protect it with that. Almost?
In some ways, yeah.
So it's almost like an insect in a gall.
Yeah. I think that's a really good way to think.
Like a
insect in a
parasitic wasp or something that injects something into a leaf, and then you see the galls on the leaves or into another caterpillar or something like that.
Yeah. So they're affecting their host's physiology in a way that it's actually growing to protect it.
Interesting.
So I think that's a really good analogy.
Yeah, it's like a gull, like an underwater gull. Like a living.
You really like that idea.
I love that idea. Well, I really like the idea of like a parasitic, like parasitic wasps are so cool.
Yeah, they are.
Yeah.
And I make my first year students learn about them.
They're so cool. But that's really kind of almost similar.
It's similar in a lot of ways.
Yeah.
And so but they also have a crazy behavior too. Like most mussels, when you encounter them in the river, they sit up.
Okay.
Right? So they have these little siphons that pull water through, and they're sitting up in the water column. They stick their foot out, and they turn on their side.
These are the rainbow.
The rainbow
mussels.
mussels.
Rainbow mussels.
And again, these are ones that I know from talking to people who've worked with them. I desperately want to see one displaying.
So you haven't seen one displaying yet?
No.
Oh,
much to my shame.
Todd Levine life goal. See,
right, this is my life list.
Whoever's out there, whoever will ever listen to this, take Todd out to see a rainbow mussel displaying.
I mean, I've ticked off some things. I saw a sundew in the wild. I'm proud of that. It's actually not that hard here.
Don't even talk to me about sundews. I've tried. I went to Peninsula.
I can fix this. If you can fix the rainbow for me, I can fix the sundew for you.
I went to the Ridges in Door County, which is known to have sundew.
Mm-hmm.
And I went there 3 weekends in a row.
Oh, ouch.
And my parents, like, they have a condo up there, so like I would go up there every weekend and I would look through the ridges and follow all these paths and try and find sundews. Never found one. I found a bunch of pitcher plants, which were very cool to see.
Are we friends on Facebook?
I think so.
I don't know if we are or not.
Oh, wow.
But I have sundews from this summer.
Oh, really?
Yeah.
So I think my issue with looking for sundews is I didn't know the scale of plant that I was trying to look for. And so, you know what I mean? Like,
yeah, I do.
If you don't know what you're looking for, it's hard to look for it.
Oh, it is. Yeah, you're right. You need a...
It's like if you're ever looking for a rainbow mussel and you're wondering if it's like this big or this big.
And it's closer to the second one.
Yeah, you're going to look for a little lure then. So you know. Yeah.
Oh, man, I really want to see if... I hope my sundew's on here.
Have you ever wondered if you were like snorkeling and looking for rainbow mussels and just thought it was a crayfish? Like it tricked you?
You know, I don't think their lures are that good.
Okay.
But the fish lures are that good.
Yeah, okay.
But it's the behavior that actually does the rainbows, that makes the rainbows so extraordinary. Because they're sticking out their foot, they turn on their side, and then not only do they wave the little tentacle arm leg
like the
things,
claws?
Not claws, but more like their walking legs.
Really?
Crayfish. Yeah.
So they actually have that many appendages?
Oh, yeah.
Because I assumed this was like a fishing lure where it was like just like the claws and then like the body.
I don't know if they have the same number of legs as a crayfish does. But right, it
yeah,
doesn't matter.
I mean, that's amazing either way.
Right,
That they even
they
have
can't see.
Separate
they
legs.
can't choose to grow in this way.
That is nuts.
Whoops, there you
Thanks.
go. Yeah, I didn't do a very good job.
That's good.
But yeah, I feel like you need to see this.
Yeah, seriously.
But yeah, it's shocking. Like, really super amazing. You're gonna be amazed
At the sundew?
Oh.
Are you showing me? What are you showing me right now?
No, I'm about
showing
to
me
show you a rainbow mussel.
Okay, I'm ready for it. So one question that I wrote down earlier because I just couldn't shake it.
Okay. Go for it.
So I was thinking from like an ecological standpoint, and I'm wondering. Oh, is this the video?
This is the video.
All right. Oh my god!
Right?
What? So it puts its foot down and actually moves its shell.
Yes.
It moves its whole...
And it does the escape behavior.
It's like an active living animal. This is like an active living...
they're all active living animals.
Well, but you think about... All right. I'm sorry. I'm used to working with like zebra mussels, which they like sit there and like...
Right,
You know, that's all they do.
But these are our native mussels. These are the ones we have to protect.
That was nuts, man.
Right?
So that thing is like...
And they're doing They're doing an escape
crawling
behavior.
around.
Like not only are these mussels are flipped out on their sides where mussels don't normally hang out.
Yeah.
But they're flapping these little leggy things.
Yeah.
And they're moving backwards towards this fake tail.
Yeah. That is crazy.
They have this whole suite of behaviors.
And these things don't have an eye. They have never seen a crayfish.
Nope, never seen a crayfish.
They don't have the thought to see like they've literally had to somehow adapt to know.
Yeah.
That is amazing.
Yeah, it's incredible.
So then a fish comes along and is like, "Oh, that's a tasty morsel.
Right.
I'm gonna bite it." And so they bite it, and
you look delicious.
And then they release their. And then what happens then? So then the fish swims around, obviously, because that's what fish do. These things kind of get nice and protected by whatever antibodies the fish is creating.
And they do use some of the resources on the fish.
So they are a little bit parasitic.
There's a lab in Kentucky who've actually grown Glochidia with fish blood and agar.
Okay.
So we know that the mussels are getting something while they're on the fish.
They're getting nutrients.
Right. And there have been at times when I've thought and other people have thought that the mussels are hitching a ride.
Yeah, just trying to get upstream.
They're not getting that much.
Well, trying to spread their population.
Right. But these, it's pretty clear that they're getting something. Like I said earlier, 300 species. Some of them are getting more than others.
Sure. Yeah.
Right.
Yeah.
But they're getting a ride, but they're also getting something nutritive. And I don't think we've really explored the depth of that connection.
That's amazing. I can't believe how little it's been explored for how amazing of a activity and like a display that is. I mean, it's
and that's what struck me. Like that's why I do what I do.
Yeah,
because I've been canoeing over the top of these things. Had no idea they were there. Had no idea what they were doing. And then one day somebody told me this is happening. And then a year later, or it was 1 to 2 years later, I had the opportunity to do research on it. Really?
That's
okay.
Yeah.
Yeah, and so I went from thinking I was going to research bears and wolves and stuff, and now I'm laying on my belly in a river.
So now you're laying on your belly, got an inner tube, some floaties.
It was not in the plan.
Do you okay, so here's a question that I was thinking of before. So you were talking about these different lures, and I'm just wondering, like from an ecological standpoint, do these different mussels attract, do you think that they are evolving, I guess I'll use the word, to attract different types of fish?
Absolutely.
So one specific host is going to be...
Yeah, and we haven't really talked about the physiology, partially because that's not a lot of what I've done.
Okay.
But the way that we recognize mussel hosts right now is a little short-sighted. In the sense that a lot of what we've done, for the last 30 years or so, is we take mussel babies, the Glochidia, we take fish, put them together, and if we get young mussels out of that can live on their own, then it's a host.
So that's what happens? So
that's live in conservation.
No, I mean just...
go ahead.
So that's what happens with the mussels. So they live on the fish for a certain amount of time until they grow into strong enough to...
And they actually go through this metamorphic process.
Okay.
And they change their body structure.
Are they called instars?
No, I don't think
that's
so.
Like insects. Insects.
Right.
Instars.
Yeah, but I don't think so.
Okay.
I've never heard anybody apply it to mussel.
Okay. I don't know.
But it could be.
I'm just wondering.
I like
question,
it.
Yeah.
I like the word, and I wish we could use it in malacology.
Are there
too.
Defining stages?
But there's the Glochidia has one adductor.
Okay.
And the adult has two.
Oh, okay.
Right.
And so they grow a muscle, basically.
Yeah.
Okay.
Muscle grows a muscle.
Damn it! I didn't even realize. So the mussel has two muscles.
Yeah. And so they go off and they go from feeding on fish whatever, right? Fish blood, fish,
nutrients,
to filter feeding.
Okay.
And there's some debate about what happens once they fall off the fish. They're real hard to find. They're small. They hang out for a short period of time. And then we start seeing them after this sort of empty period.
Really?
And I haven't done a good literature search. I haven't kept up with some of the things that are going on in the field to know if something's been found in the last 5 years or so.
Okay.
But that period after they leave the fish and before they sort of come up and start filter feeding, there are a lot of question marks associated with it.
Interesting. Almost like a turion from a plant where it just kind of falls off and it kind of waits for a better time to start.
And we just don't we don't know what's going on there.
Yeah.
I mean, I think that there could be a lot of interesting things happening.
Interesting.
I'm not sure that and maybe we find out, they fall off, they start doing their thing, and then they get bigger, and we start finding.
And they just kind of start and just get
could be
bigger
as
and
simple
bigger.
as that.
Yeah.
But there have been some other interesting things that people have talked about, like maybe they're down in the sediments and they're not filter feeding, they're actually pushing things up in eating in different ways.
Interesting. Yeah.
Yeah, and
That's how they figured out what crayfish
I don't know what
look
the status is on that, but it's kind of cool, right?
Yeah, that's very cool.
Right, it's where they met their very first crayfish.
Yeah. Little crayfish are like, I should look like you. You look pretty tasty. They ate some of them and they were like,
Nice.
I don't know. Yeah. Wow. So the rainbow mussel is in just southeastern Wisconsin.
Yeah. So DNR has some nice range maps that you should probably consider using.
Okay.
To look at that, and it's just sort of in this corner of the state. The only ones I'm aware of are in the Muskogee River, although there may be some elsewhere. And we've done a lot of survey work. There are some that we may have found outside of there, but the populations that we know about are sort of hanging out in that corner.
So down in Sheboygan, Ozaki, Washington, Waukesha, Racine,
and those are
Jefferson counties,
right?
And
so
Dane counties.
This may or may not be active populations.
Okay, yeah.
These are where they've been found historically.
Okay, yeah. Cool.
Yeah.
Very cool. Dang, dude.
They're awesome animals.
Yeah, I really want to go out and see some of those.
We need to go snorkeling.
Yeah, let's go snorkeling.
Let's go
lay
right face down in the river.
Right now.
You really want to get that story out of me, don't you?
A little cool. Kind of.
Yeah, so I don't know how to segue into it. It's going to be...
I think you just need to start.
Yeah. Let's do it. So Aaron was doing some work with me, and these lures.
This story. So this is.
I assume that's what you were looking for.
Wait, is this the police?
Yeah.
The police.
Yeah.
Okay. This was with Aaron,
Yeah.
Aaron that I've met.
Yeah.
Oh, that's
that
so
Aaron.
funny. Okay.
Yeah. So one of the things that the unanswered questions here is how do the mussels use these lures to get fish to attack them?
Mm-hmm.
They're not just sitting out on the bottom of the river going flap, flap. Hey, I'm a fish.
Right.
Flap, flap all day long. So Aaron and I set out to try and understand a little bit about how mussels might have different behaviors and how those behaviors might elicit attacks from fish.
Mm-hmm.
So one of the things we did is we were trying to look at whether or not mussels had different behaviors for different times of day.
Okay.
Right? So the fish are out hunting.
Yeah.
You might want to be out at that time.
Fish are diurnals. Most of the time they're diurnal species. Right?
Yeah, and it actually looks like the fish that these guys target might be crepuscular. So
sure.
Beginning of the day, end of the day.
Yeah. Just like deer.
Yeah. So we wanted to know, when are they doing this? Are they displaying all day long? That seems kind of wasteful.
Right.
Or are they displaying it at certain targeted times? times.
Interesting. Good question.
Yeah, I thought so. And
Yeah.
a lot of other people like it too, but it's a tough one to answer. And at the end of the day, Aaron's data are good data, but they don't totally answer the question and put it away.
Okay.
Because it takes a lot to get that answer. But so we decided to work in the McGuanago, and that's where we first described these different lures. Go for it.
I was just going to say, how much does it take?
I don't know. Do you have a grant?
Okay. All right.
So we're doing these observations, and we go down to the river over and over again, like 6 a.m. to 8 p.m. And finally we decide, we would really like to know if there's a difference between crepuscular and nocturnal.
Yeah, that's a really good question.
Right.
Yeah.
So are they displaying it morning and evening, or are they doing this all night?
Right.
And
like, is
then
it
they
like an
stop it.
automatic? Because you got to wonder.
Oh,
if
you
it's
do.
just an automatic, like we do this every hour on the hour, like, or do we do this because we actually know
Right.
if it's day or light or day or night?
Right. And so we set up this whole system of checks. We ended up doing like 9 different days where we check these mussels for a couple of hours each.
Okay.
And we were trying to distribute those around the clock in different ways to make sure that we'd catch things that were happening.
Sure.
And finally we decided that we'd do an overnight set. And so we went camping and brought friends, family, whatever.
Yeah.
And
Hey,
so,
we're going to check some mussels. Let's do a camping trip.
Right, because
Yeah.
I know you really wanted to get up at 3 a.m. with me.
Right. Yeah.
So Aaron and I are taking turns overnight. She goes down to the river, and she's got like quintessential, like problematic car. Is it bright red or bright yellow? And she affectionately referred to it in possibly appropriate terms as the Tijuana taxi.
Okay.
Like, when I say that, I mean, like, she might have meant that literally, that's what it was before she bought it.
Sure.
And it has, the body of the car has matching hubcaps. And it's got a PBR sticker in the back.
Amazing. Okay. Sounds like
yeah.
a pretty good car if you're living in
It's a nice
Wisconsin.
ride.
Yeah.
It's a nice ride. So she goes down to check the mussels. And I guess the police decided that maybe she was doing something sketchy. They pulled in behind her. And,
So she's in the Tijuana taxi. And she's
on a riverbank
going
at 3 o'clock in the morning.
Yeah, going just driving straight and then deciding I'm gonna stop at the river.
Yes.
Because usually at 3 a.m, stopping at the river means
she looks
something...
like she's driving to Walmart.
Right.
But she pulls off.
Usually stopping at the river means something bad's about to happen.
Right.
Especially
right.
at 3 a.m.
Yeah, a lot of normal people, and I'm not saying we're normal people.
Yeah, but they don't do that.
Right,
Yeah.
right. Normal people don't usually pull up to the river at
Yeah.
3 a.m. And somebody pulls in behind her and flips on the lights.
Okay, so it's a cop.
And nothing happens for a while, I guess. He's called backup.
Did she stay in her car or did she go to survey?
No, she stayed in the car.
Okay.
She knew. And another one pulls up, and two different jurisdictions.
So another one comes up?
Oh,
and
they totally boxed her in.
That means like they think something's going on. That's like.
And do you blame them,
serious.
really?
Well, no. Look at her car. I mean,
look at the time.
Yeah.
They box her in, and they come up. What do you think you're doing in the river? And the best quote that I've heard from any of my students. She folds open her data book, holds it up like a shield. Science? And I wasn't there. Which more's the pity. I was taking my turn to sleep. And I wish I had been there to see this. Data book? Science?
That is amazing. Oh my god. So what happened?
Everything turned out okay.
Yeah.
She eventually did convince them that she was doing science.
Okay.
It took a while.
How many breathalyzers did it take?
I didn't ask. I don't know. What I do know is that by 4 a.m. I was in the police station explaining as well.
Oh, wow. So you actually got called in.
No. She came back and was like, they let me go, but I don't know if we should keep doing this.
Yeah.
And so I went in and explained, and it was fine. It was fine. The only real negative thing to come out of it was, don't you think you should have told us?
Yeah.
If you were going to be sitting on the riverbank in the middle of the night.
Word for anybody who would be listening, tell your local jurisdiction.
Right.
Police
it's not that
force
hard.
that you're doing science, so you don't
They've
need
got
to
a
use
hard
it as
job.
a defense.
Make it a little bit easier. Share with them your plan. Share with them your research plans, right?
That's amazing, though. That's such a good story. You're just camping out.
I love this story.
Science. It's the best. Oh my god. Well, dude, I think we're getting to the end.
I think so.
But all right, so a couple of things before we end.
Yeah.
Are there any research publications that you want to promote? Anything you're working on or that you want to work on? You kind of mentioned some earlier.
Yeah, I think that really a couple of things
for young scientists to think about too.
Okay.
You know, like what can be.
So first, explore your I mean, as far as like what young scientists should think about, explore your passions. Like, I was really boxed into the large mammals thing.
Mm-hmm.
And when I learned that I'd been canoeing over these things, it blew my mind. And it completely changed the way I went.
Yeah.
And it very nearly happened more than once with more than one set of organisms. Like when I learned about algae that glowed and made toxins and that they're different and they're not, that blew my mind too. And so I think just stay open to having different options and different experiences, because you can make a difference, and you've got to figure out what drives you.
Mm-hmm.
Like, do you want to make sure that species survives? Do you want to understand the way life works? There are a lot of legitimate and important questions that we can ask, and none of them are necessarily more important than another. I think it's really hard for us to rank them.
Mm-hmm.
But it's real easy for outside people to look at it and say, this one's important, that one's not.
Right.
And I think in a lot of cases, we really need to be humble about what we know and what we don't know.
Mm-hmm.
And in a lot of cases, we don't know enough to rank. This is the important question. That's not the important
100%.
one.
Well said. Yeah.
Thanks.
Yeah.
Appreciate it.
Yeah.
But I think for young scientists, or young... people do that, right?
Young people, yeah.
Yeah, for anybody really, just stay open. Don't plan too hard. Let yourself be open to those opportunities. Like, wow, there's a whole new taxon that I didn't know about.
Like, latch onto something if you want, right? But
explore it.
But don't be so...
Explore it, but don't get stuck in it.
Right. Yeah, like allow yourself to be open to like new opportunities.
Yeah, and I think
and whatever fascinates you.
With my job now, that's a really important thing for my students.
Mm-hmm.
And my forcing myself to be willing to open up to those new opportunities is really important for them. I've started working with these crazy little crustaceans that live and die. They lay their eggs, and then their eggs dry out completely. So we literally have next year's population in a Ziploc baggie on the shelf.
That's amazing.
Yeah, and so...
What are they?
They're triops.
Water bears?
Nope.
No, triops, like the desert.
Triops are tadpole shrimp.
Yeah.
Yeah, so I think that was a cool opportunity for my students. And we're working with fairy shrimp and...
Fairy shrimp.
Yeah. So like just being open, even for me, I have to force myself to be open to think, there are these other organisms I can work with. And that might be good for me. That might be good for my students. That might be good for science.
Mm-hmm.
And I think that openness is really important. So what was the other questions you asked?
No, that was a really good one.
Awesome.
Actually, that kind of answered the last question, which is if you could tell your high school or college age self some advice, what would it be?
Yeah.
And that makes a lot of sense. Like keep an open mind and
yeah. And I guess as far as opportunities or papers or whatever,
that's
like there are things I'm proud of,
Yeah.
but really I'm looking to the future, and the two big things are I desperately want to understand some of the genetics behind what's going on in that population of mussels with the different lures.
Yeah.
That seems like a really cool thing that we could be doing. The other thing is that I'm starting to work more and more with citizen science.
Okay.
And I've got a project through the Milwaukee Public Museum that we're trying to go public with through what's called the Zooniverse platform.
Yes, I've heard about the Zooniverse platform.
Ah, nice.
It's very cool.
You should beta test this.
I think I have actually a little bit.
Oh, you have,
collect
haven't you?
mussels, right?
Yes.
And then you put their dimensions into the.
So you measure museum specimens.
Yeah.
And what we're trying to do is to find out whether or not the environment is changing the shape of our mussels.
Okay.
Like, so as we convert something from forest to agriculture, or we go back and we plant some oak trees, or we change the tillage practices. All those are things that change what's at the bottom of the hill, what's in the watershed.
Yeah.
Right? And so what we're trying to do is get these museum records from like 1900 and 1970 and that I collected in 2016 and put them all on the same map and ask whether or not the dimensions within a species are changing. Are mussels getting bigger and heavier? Are they getting smaller and longer? How are they changing in response to the way that their environment's changing?
Yeah. So what can you do as a citizen scientist?
So as a citizen scientist, right now, what you can do is help us to collect data on those shells.
So are there shells from the museum?
Absolutely.
Okay.
Yeah, and so you don't actually even have to go to the museum.
Really?
You can log in on Zooniverse.
What's the website? Just Zooniverse.com?
So it's zooniverse.org.
Okay.
And we're not an official Zooniverse project, so you'd have to contact me, but it's publicly available.
How do you contact you?
You can email me at Carroll University, Levine, Levine, at carrollu.edu. And you can come in and check those out and
very cool.
And add your data.
That's super cool. So they'll literally send you a box of mussels to measure.
Well, no, actually we're digitizing the collection.
Oh.
So I'm working with museum staff who are in charge of getting pictures of every mussel in their collection.
Okay.
So this isn't the stuff that you walk in and pay to see. This is the stuff behind the scenes.
Yeah, like total stuff that's back on shelves.
And the whole reason for a museum like that to exist is so that we, the big wide we,
the scientific community,
not only the scientific community, but the
public
public too, right?
Yeah.
Like, so that we can reflect on our natural world.
Yeah.
Like, the Milwaukee Public Museum is trying to position itself to be the Natural History Museum of Wisconsin. And what that means is that they're literally a public service for all of us to help us understand where Wisconsin came from and where it's going. And I'm hoping that my project gets to be one of the first, one of the public faces of that, and to help us in a very transparent way look at our environment and ask, where did we come from 108 years ago?
Yeah. And
where do we want that to go?
Totally. That's very cool, man. Well,
thanks.
That's awesome. So TLEVINE at Carroll, CAROLL.
2 R's, 2 L's.
Oh, okay. So why don't you spell it out?
T Levine, TLEVINE at CarrollU, CARROLLU.edu.
Perfect.
The other alternative is Gmail, which is Levine, LEVINE, TD at gmail.com.
Okay. And you can send people info about mussel measurement.
Be happy to.
Awesome. Well, Todd, it was great to have you on the first ever podcast of Wisconservation. Awesome. I think it went really well. That was super cool. So
nice.
Yeah, I'm
this
really
is
stoked.
Fun.
I think this is gonna be a good thing.
I agree. Cool. I'm excited you're doing this.
Yeah, me too, man. I'm excited to have you on here and just talk about your stuff, and it's so fun to get nerdy with you. I don't know. It's like so.
Yeah.
It's so fun. Cool. All right. Well,
Had a great time, too.
I'm gonna turn this off.
Sounds good.